Last class we talked about proteins, the peptide bond between amino acids, and then the bonds within the protein molecules. There are three bonds that are highlighted: hydrogen bonds, disulfide bonds, and ionic bonds. Today we will talk about protein structure and how, right, not the process of proteins being made from DNA, but how the amino acids are put into sequence. Well, not how, but the fact that they are put into sequence, and then how the polypeptide chain is folded into compact structures. So the topic is protein structure. And there are four levels of organization for any protein. And keep in mind, protein could mean a single polypeptide chain. It could mean multiple polypeptide chains that come together to form a protein. So it really depends on what the protein is made, what in the sense at how many polypeptide chains the protein is made up of. So there are four levels of organization. Obviously, starting with the very first one, the primary structure. The primary structure is simply the sequence of amino acids. In the polypeptide chain. Now we know that that sequence is determined by the sequence of bases in the DNA, in the gene. So, right, you have only 20 amino acids, right? And a polypeptide chain can be, well, we saw yesterday that insulin is an example of a very small polypeptide chain, only 51 amino acids, but in general they tend to be between 100 or a few hundreds, right, up to 1,000. So even if you have like a chain with 100 of them, the sequence can be different. And right, even if you take a look at, for example, 6 of these, right, so you've got these are 6 amino acids. So these are the codes for the amino acids. You don't really need to know the names of each and every one of them, although, right, it is something that you can easily find. If I take you, right, to an index of amino acids, so just let me share that on screen. Alright, can you see that?
Yes, sir.
This is from the Cambridge book, by the way. Alright, so you can see these are the 20 amino acids, their full names and their 3-letter abbreviation, and their structure as well. So you can see, right, the brown block represents the general structure, which is the NH2 group, the C double bond OH, the carboxyl group, the carbon and the hydrogen. So that's common, right? So what they're highlighting is basically the R groups. And you can see that they are simpler ones. They're straight chains with nitrogen, double bonds, branching, and you've got cysteine over here, which forms the disulfide bonds. You've got ring structures, right, as well, and something like proline. So there's a lot of variety. And the ones I've put in on the whiteboard are valine, and then there's glue, so glutamic acid, so that's glue. Then histamine, histidine. Histamine is related to the immune system and allergies, so histidine. Leucine up here, and cysteine, and finally glycine. Right, now I put those in randomly, right? It's not, I mean, it could be a protein that starts with those 6 amino acids. I've put them in randomly, just an example. So going back to the whiteboard, let me change screens. Right, and so you can imagine the combinations that you can have with these, right? If you just keep the first amino acid, right, the same, you keep it constant, and then you move these guys around. So you've got histine, and then glue, histidine, and glutamic acid, and leucine, and cysteine, and glycine. And then you got these two, so you bring in leucine first, and then glutamic acid, and then histidine, and cysteine, and glycine, and you get the idea, right? So even if you've got 6 to start off with, there's so many variations in the sequence that can occur, and and this is only for me maintaining the first one and then moving the rest around, right? I can then do the same for each one of these in the positions they are have, and I can come up, I can end up with so many, right? There's like you can work out the number of combinations that are possible. And so the sequence of amino acids, the primary structure is very important, and we will learn later that if the primary structure is somehow disrupted due to mutations in the DNA sequence, then that can result in faulty proteins, right? And that normally, I mean, it could result in nothing happening, possibly, right? It could also result in mutations. It could result in faulty proteins that are non-functional, and then obviously that will have a detrimental effect. It will cause some sort of disease or some sort of deficiency, because whichever protein is affected won't be able to be produced, and that particular bodily function will be impaired, right, to some extent or entirely. So it's easy to say that it's just the sequence of amino acids, but we can understand how important that sequence is. Next up, secondary structure. By the way, you asked me for the Cambridge textbook. Did I come around to sharing it with you or not?
No, sir.
Okay, my bad. I'll do that. Okay. I just recalled, it slipped my mind, so I'll share that with you. Okay. The secondary structure is a in a protein is, you know, the coiling or the arrangement of the polypeptide chain into a regular pattern, into a regular shape. So your polypeptide chain is formed over here by the sequence. So that's right, you have a polypeptide chain at step 1. And so the arrangement, the 3-dimensional arrangement of that polypeptide chain into a regular shape. And there are 2 types of shapes that we have. One is called the alpha helix. Alright, so this is a right-handed helix. And it is such that it forms a coil. So right, that's what it looks like. And you've got the peptide bonds that form the backbone of peptide bonds form the backbone. And the R groups are sticking out, not in. Right? Obviously, you got many more than those. So the R groups are sticking out of the helix. And the helix is held in place, so the shape is held in place by hydrogen bonds between the amine and carboxyl groups of the amino acids. So remember yesterday when we were talking about hydrogen bonds, I mentioned that the bonds can occur between R groups and also between the amine and the carboxyl groups. Alright, so in the secondary structure, it is held in place by hydrogen bonds, but these hydrogen bonds are being formed between the amine and carboxyl groups of the amino acids. The other type of secondary structure is called beta sheets, right, or beta pleated sheets. And so as the name suggests, the polypeptide chain forms a sheet something like that. And so if that is one sheet, you've got another one coming along like this. And then you've got again, right, so a single sheet, right, is just folded in a particular way, right. Multiple sheets, if they align together, those multiple sheets are held together by hydrogen bonds. Alright, so the polypeptide chain folds into pleated sheets. In fact, I'd probably remove that. And right, multiple sheets are held together by hydrogen bonds. And again, these hydrogen bonds are the ones occurring between the carboxyl and amine groups of the amino acids. Now, one thing to keep in mind that alpha helices, well, beta pleated sheet in fact, right, are common in fibrous proteins, and we'll get to what fibrous and globular proteins are. So it's a common secondary structure in fibrous proteins. And when we talk about fibrous proteins, we will understand that these are, right, they contain a majority of hydrophobic amino acids. Right, what determines whether an amino acid is hydrophobic or hydrophilic? The R group. Right, so if the R group has some polarity to it, then it will be hydrophilic. Hydrophobic. So if there's some polarity in the R group, it will be hydrophilic, which means that it can, be a solute in an aqueous medium, which basically means the blood and tissue fluid. If it's hydrophobic, then it would, and we'll see the structures. So those are the structures that are generally impermeable to water, right? And that makes sense because they don't like water, they're hydrophobic, so they repel water. But again, we will come to that very briefly, very shortly. Now, the third level is the tertiary structure. Oh, by the way, as I said, the alpha helix and the beta sheet are the common secondary structure. Sometimes proteins, sometimes they just undergo random folding and coiling. Alright, so they don't have a proper three-dimensional arrangement such as an alpha helix or a beta pleated sheet. Now, the tertiary structure. The tertiary structure is where interactions between R groups causes, right, further folding or coiling of the polypeptide chain. And in this case, there's no particular shape to highlight, but right, the R groups are involved, and so we know that you either have hydrogen bonds, right, ionic bonds, or disulfide bonds happening. And so, right, it is the tertiary structure that is the level of organization at which the R groups become involved in the structure of the protein. And then finally you have the quaternary structure. And this structure is only applicable to proteins that are made up of more than one polypeptide chain. And so over here again the R groups are involved, and multiple, depending on the protein obviously, multiple polypeptide chains can come together, and there's interaction between the R groups, and they fold and fit together in a particular shape. R group interactions. An example of this is hemoglobin. So we are familiar with hemoglobin. It is the protein that is in the red blood cells to which oxygen binds, and this guy is made up of 4 polypeptide chains. Right, there are 2 alpha chains, not going into details, 2 alpha chains, 2 beta chains. Do not confuse the alpha chain and the beta chain over here with the alpha helix and the beta pleated sheet, right? So that's different. This is the name of this chain given to the polypeptide chains, their length and coiling and all that, right? So 2 alpha chains, 2 beta chains over here, and 4 in total, and so those 4 come together to form one molecule of the hemoglobin protein, right? So that is an example of a quaternary structure. Examples of tertiary structures are, the fibrous and globular proteins. Well, again, it's just a higher level of folding and coiling. So fibrous and globular is more about. It is obviously related to the tertiary structure, about how the R groups are arranged. Okay, any questions so far?
None, sir.
Now, one thing to just keep in mind is that we've learned the different types of bonds that hold the protein together at different levels of organization, and so we should understand why proteins in general are sensitive to temperature and pH. Right, and that is because of all the interactions, all the bonds within the protein molecule, whether that's happening between the amine and carboxyl groups of the amino acid molecules or between the R groups. So for example, right, the hydrogen bonds, they are affected by pH, well, temperature as well, right? So if the temperature changes too much, right, if there's a slight change, then and we'll look at this more in the next chapter, which is about enzymes. But right, if there's a slight, proteins have an optimum, right, pH and temperature range at which they work. So a slight change will affect their activity and efficiency. But if the change is drastic, and again drastic, I'll put the inverted commas because it really depends on the protein how sensitive it is, then the protein will end up, right, these bonds will be affected and the protein molecule will end up losing its shape because those bonds will be broken. It will become D-shaped. Actually, not can but will. It will lose its shape and it will become, it will lose its activity. And at this point, a protein is said to be denatured. Right, denaturation has occurred. Denaturation has occurred, and the protein is said to be denatured. So it's lost activity, and there's no going back. Right, once this happens, right, it is not reversible. So you cannot take up, you can't put it back at its proper pH. I mean, you can, right, but it's not going to fix it. It's not going to help reform the bonds. That's, that particular molecule is done, right? Now, let's talk about fibrous and globular proteins. Starting off with fibrous proteins. Right, so fibrous and globular proteins are it's a categorization of proteins as to, right, their folding, so their structure. So this talks about the structure and also their role. So as you know, right, fibrous proteins are, a lot of them have beta pleated sheets, and they contain a high percentage or proportion of hydrophobic amino acids, or hydrophobic R groups to be more specific. And because they're hydrophobic, you'll be able to find fibrous proteins in structures such as your hair and nails and skin, and all of these are, they are waterproof structures, and or you could say they're impermeable to water. Right, so your hair doesn't sit well with water, your skin, your nails doesn't allow water to enter, right, it doesn't allow water to pass through, so they're waterproof, and that's because they have a lot of hydrophobic R groups in the polypeptide chain in the amino acids, and it just happens that, right, the structure that they form is fibrous protein. So what you have over here is, right, long parallel polypeptide chains with cross links to form fibers. And fibrous proteins, again, they're insoluble and they're tough. And they play, they're suited and they play a role in structural functions. So for example, right, connective tissues in, that cover up the bones and connect your bones to your muscles. An example is collagen, right? So they've detailed collagen as an example. And perhaps you've heard of this. And it's sort of on a side note, it's funny if you look at companies that deal with beauty products, and they tend to throw around scientific terms. Maybe those are names of proteins, maybe they're names of amino acids, maybe they're names of some other biological molecule, and they're like, oh, it's going to help this and it's going to improve that. And if you know the reality, if you actually know the science behind the term that they're using, you realize that what they're saying is absolute nonsense, right? They're just using those fancy words to sound impressive. And collagen is one of the proteins that these companies use. Like, oh, it's going to improve your collagen and stuff like that, or take collagen supplements or whatever. Okay, so collagen is a fibrous protein, and it is so abundant, right? Up to 35% of the protein in your body is collagen. So it makes up to 35% of the protein in the body. Obviously that's a huge chunk. And it is, it's a protein that gives strength to tendons, ligaments, bones and skin. Now they talk about the structure. It has a high tensile strength, right? And they talk about the structure. So this guy has, fibrous proteins generally don't go beyond a secondary structure. Alright, so if I just add that to the previous page, do I have space? So you generally don't go beyond a secondary structure. Generally, but collagen has a tertiary structure, so in fact quaternary structure. And the quaternary structure involves 3 polypeptide chains. That wind around each other in a helix. And so what it looks like is perhaps that's 1, and then that's 2, and so you've got 3 polypeptide chains if the colors are visible, and they wind around each other into a helix, and they're held together by a large number of hydrogen bonds. Right, and so these guys, right, when they are associated with, combined with bones to give them strength, right, so the strengthened bones and the analogy they use is how right, steel is used with cement to make reinforced concrete. So that's the analogy used of how. And then they mention that there's a disease where the collagen fibers do not properly develop, and so the bones become weak and brittle and they break easily. Okay, also just one minor detail, the primary structure of the polypeptide chains in collagen is a repeating sequence of glycine, as every third amino acid. They mention that it's a sequence of 3, and the other 2 also tend to be proline and hydroxyproline, but not always, so that's a tendency of repetition, but every third amino acid is glycine. Okay, now to quickly, I want to wrap up this chapter, so quickly moving on to globular proteins. Any questions about collagen and fibrous proteins?
No, sir.
So globular proteins have a mix of hydrophobic and hydrophilic R groups. And so what happens is, right, globular means they tend to form spheres. And so the hydrophobic R groups fold in the polypeptide chain folds in such a way that the hydrophobic R groups are on the inside, and that keeps them away from water or the aqueous environment. And the hydrophilic R groups are on the R side, and that helps the protein be a solute in the aqueous environment. Okay. Now what happens is, right, I'm not using the word dissolve. These guys don't dissolve, but they sort of form a suspension in the aqueous environment. A suspension of particles, right, of protein molecules, and the term for that is a colloid. Right, so it's not dissolved like sugar would dissolve or salt would dissolve in water. It's not dissolved, it is suspended uniformly throughout, and they don't settle, but right, it also makes them difficult to remove. And examples of globular proteins we've seen, right, hemoglobin is a globular protein. There's also the antibodies that are produced by the immune system. Right, those are examples of colloids and right, globular proteins. All enzymes are globular. Some hormones. Right, yeah, and finally, right, there's something called conjugated proteins. So that's the last bit of this chapter. And so basically what this means is, right, you've got a protein molecule plus, right, another molecule or substance that is non-protein, that is attached to the protein, which is called a prosthetic group. Right, so it is a non-protein molecule or substance, right, that is attached to the protein. Examples, right, are hemoglobin, right, because hemoglobin has a heme group which has iron in it, that obviously is involved in the binding of oxygen and carrying of oxygen, right, but because it is a non-protein molecule that is associated with the protein, and so that is an example of a prosthetic. The iron is the heme group, and the iron in the heme group is an example of the prosthetic group, and that makes hemoglobin a conjugated protein. Then general terms such as lipoproteins, which, as the name suggests, are protein molecules with conjugated lipids. Right, and you can find them, for example, right, proteins in the cell surface membrane or the cell membrane. Right, they could be present as antigens, they could be present as receptors, or obviously they have some function. Another example of lipoproteins is cholesterol. The proteins present in the blood that transport cholesterol. Those are called LDL, which is low density lipoprotein. You might have heard of these. And HDL, which is high density lipoprotein. Right, and right, HDL contains more protein than lipid. So that's why it has a higher density as compared to the LDL. It contains the proportion of protein to the lipid in the LDL is lesser. So I just squeeze that in over there. Lesser proportion of protein to lipid. Okay. And then you've got glycoproteins. And as the name suggests, right, you've got a prosthetic group attached to the protein, which is a carbohydrate. So and what the carbohydrate molecule helps them to do is, right, it retains a lot of water and makes it more difficult to be digested by proteins. So the carbohydrate, right, retains a lot of water and makes digesting it difficult. And examples of glycoproteins are mucus, right? So mucus, we are very well aware, is very it has a lot of water content. And another example is the synovial fluid, which is present in the joints of the body. Right, and so it allows for lubrication. And mucus is not only for what is discharged from our nose or from our throat during infection. It's present in our GI tract all the way through, and helps lubricate the esophagus and the other parts of the GI tract for food to move through. And the fact that it's difficult to digest, right? So you've got a mucus lining the walls of the stomach, and that helps, right? So it's difficult to digest, and so it helps to protect the cells of the wall from the stomach acid. Right, and we are done. Okay, if you have just a minute, a very last thing. Right, you take something that is called a biuret reagent, right, and you mix it with a sample that contains protein. Well, presumably, right, and so it's given in the book. So the biuret reagent is blue in color, and if protein is present, right, the color turns purple. No heating is involved. That's all you have to do. Okay? Any questions?
Awesome.
Okay, and that brings us to the end of the chapter. And right, so that we're done with biological molecules. The next time we will start with the chapter of enzymes. All right?
2B.
Yeah.
Yeah.
Right. So that is. Let me just. It's page 100 and something.
Yeah, it's 102.
Yeah. So 2B1 is what we'll
Yeah.
be going for. Okay.
Okay.
All right. See you next time, inshallah.
Yeah. Love this.
Allah.